A DNA can be isolated from a number of plant tissues. Young leaf tissue is a good source of DNA because young cells are gradually dividing and they are having the fresh DNA. Leaf tissue is placed in the tube and frozen using liquid nitrogen. The freezing helps the mechanical breaks of the cell. A tool resembling a drill is used to pulverize the tissue into fine powder. It's just like a homogenizer, or just like your day-to-day life utilizing mixture machine. A solution containing buffer, salts and detergent is added to the tissue. The buffer and salt help stabilize the DNA while the detergent breaks the cell membrane. Heat helps to disrupt the cell, so the tissue is incubated at 60 degrees Celsius temperature with frequent mixing. During incubation, the contents of the cell are released into solution. This not only includes the DNA but also other cellular components that must be separated from the DNA. Chloroform, which is a solvent, is added to wash away the unwanted cell components, such as different proteins and different ingredients inside the cell. Because chloroform is an organic solvent, it can solubilize many organic particles. The sample is thoroughly mixed. The sample is spun very rapidly in a centrifuge. Centrifugal force separates the solvent layer from the DNA solution. Now this is the DNA solution is separating from the chloroform with unwanted cellular components. The DNA layer is removed to clean the tube. Cold ethanol is layered on the top of the DNA solution. When the DNA solution is mixed with the ethanol, then the DNA precipitates. It comes out of the solution. The sample is centrifuged and the solution is removed, so the only DNA remaining at this tube. The pure DNA is dissolved and used in molecular techniques like PCR and other techniques to study the biological activities of the plant cell.
Once DNA has been extracted from a sample and a section bearing useful code such as a BNTR or STR is isolated, the next step in DNA fingerprinting is to measure each strand and count the number of repeated sections. To do this, scientists use a technique called gel electrophoresis, which uses an electric current to push strands of DNA through a slab of gel-like material. Because each bit of DNA is negatively charged and subject to an equal electric force propelling it to the positively charged side of the gel, smaller pieces move faster than larger ones. When the current is removed, the gel is photographed to reveal how far each bit has migrated. By comparing the bands produced by the DNA samples of interest with bands produced by standard samples whose sizes are already known, the precise length of each DNA fragment can be gauged.
A sample of chromosomal DNA, also called the genomic DNA, can be used as the starting material for the polymerase chain reaction, also known as PCR. With PCR, an investigator can amplify a single copy of a DNA segment into billions of identical copies. The DNA segment of interest or target DNA is indicated as red. In addition to the target DNA, a PCR reaction contains several other ingredients. These include free nucleotides, DNA primers, and the enzyme Taq polymerase. The primers are typically about 20 nucleotides long and are complementary in the sequence to the ends of the target DNA. Taq DNA polymerase is delivered from hot springs bacteria and can tolerate intense heat for the PCR reaction. A PCR reaction lasts several hours and typically consists of 20 to 30 repeating cycles. A cycle begins by heating the reaction mixture to 95 degrees Celsius. The heat denatures the DNA strands, breaking the hydrogen bonds that hold the strands together. After denaturing the DNA, the temperature is reduced to around 60 degrees so that the primers can form hydrogen bonds or anneal with their complementary sequence in the target DNA. Note that the primers and the target DNA follow base pairing rules. An adenine pairs with the thymine and a cytosine pairs with the guanine. In the next phase, the temperature is raised to 72 degrees Celsius. A Taq polymerase functions optimally at the temperature and begins to polymerize, adding nucleotides to the 3' ends of each primers attached to the DNA strand. After one complete cycle, there are two double-stranded copies of the target DNA formed. The PCR reaction mixture contains mainly copies of the primers and an abundant supply of nucleotides to perform many additional cycles. After a second cycle, there are 4 copies of the target DNA produced. After cycle 3 is finished, there are 8 copies of the double-stranded target DNA sequence, and note that only 2 of the double-stranded copies consist of just the target fragment, and the others also including flanking DNA regions. As the number of cycles increases, the products consist of greater proportion of the fragments with just the target DNA increases. After 4 cycles, half of the fragment consists of just target DNA and half of the fragments also contain flanking DNA. With each additional cycle, the number of copies of our target sequence doubles. At the end of 25 cycle, there are more than 33 million copies of this double-stranded target region found.